So, good evening, everyone. My name is James Paul, and today I'm sharing my screen for the critical appraisal of liver-free plasma proteomics, identifying a haptoglobin protein as candidate marker of idiopathic pulmonary fibrosis and dysregulation of complement and oxidative pathways. My name is James Paul. I am currently pursuing my Doctor of Pharmacy from the Department of Pharmacy Practice, ISO College of Pharmacy, Nova, and Punjab. So originally, I got my article as such. I'll show you my article. So this is my first manuscript, which I'm going to show, which is of it's named as Label-Free Plasma Proteomics, identifying as haptoglobin-related protein as candidate marker of idiopathic pulmonary fibrosis and dysregulation of complement. So in short to say that they are haptoglobin-related markers, new haptoglobin markers which have been identified as candidate markers for the idiopathic, which is not known nature of the pulmonary fibrosis, and which is also dysregulating the oxidative pathways, which usually happens. So now coming back to my presentation. So the main author of the paper is Mike Sarswat, and his contribution is that he did an extensive research in proteomics as well as biomarker discovery, and his specialized focus is on respiratory diseases. Now let's go with the background literature. Background says that the idiopathic pulmonary fibrosis is a chronic and progressive lung disease, which if it's early and accurate diagnosis is critical for treatment. So for example, this is idiopathic in nature, so which means that its etiologies it are not known and it can happen anywhere, anytime. So it's early and accurate diagnosis very critical in giving it effective treatment. And one of the major studies is the proteomic studies, which can identify novel markers, which can say that yeah, idiopathic pulmonary fibrosis is happening. And one of those protein biomarkers is our own haptoglobin. Haptoglobins, what are haptoglobins? These are new globulins. Is a globin and hapto means four, more than five, and globins. The new related markers, protein markers in the bloodstream, which can identify due to proteome analysis and helps. So let us go with the objective of the paper. So the objective of the paper describes it as label-free plasma proteins to identify, and the focus is to identify that to serve as a biomarker for understanding dysregulations in complement and oxidative pathways. So in the complementation pathway and then the oxidative pathways, these both pathways are inflammatory pathway, and whenever the what we say fibrosis occurs, these are pathways which are activated as soon as possible, and that's why those proteins which work into activating these pathways are very essential in getting to know the diagnosis. So let's say any day due to unknown reasons, uh, pulmonary fibrosis occurs. So then these markers will very much help us because complementary pathway, complementations as well as oxygen pathway activated as soon as possible whenever the inflammation or oxidative stress occurs. Now the methodology of the paper. So what they did was they took 36 what we say blood samples, and they were identifying the particular markers which are haptoglobins. These new markers, and they used technique like volcano plot, hierarchical clustering, partially square disk analysis, injury pathways analysis. These are what I say. These are tech analysis part, and as a machines they must have used let's say proteomic analysis machines, HPLC, and etc. So, and they were going for label-free plasma proteomics, means the proteomics which have not been identified or are being novel. That's why this is very new method, because these are not even labeled by any place, and these guys used it and use very advanced statistical analysis to identify. So they themselves say because the volcano plot as well as hierarchy clustering, as well as partially square. These are very advanced techniques, and they're only used when there's huge number of data, and we have to identify whether this marker is certainly the one which is working out. So yeah, they use very good method as well as identifying those proteins. And what do they get? Let me show the paper where the result is very well identified. So this is a paper, and this is the hierarchy clustering heat map. So if you see the red, these are the classes IPF and 4. So here we have 4 as these are the markers which are coming up. And they did, I think they use meta-analysis for clustering. So there again in the paper I was talking about these clustering methods, hierarchy clustering methods, volcano plots. So they used first there is centrifuge and got the uh, samples, and they did the clustering volcano plots. This is a clustering plot heat map, clustering heat map, and it very well identified. These are the red ones. These red ones, which mean that these markers are very well present. Uh, Let me show you that. Yeah, these markers are very well present, and yeah, the partial display. So again you can see the control group and the our what we say the sample group. So these guys have and the control group, and wherever the meeting is done, which says that it is similar, and we can say that yeah, these are happening. These are the ones which are activating the complementary auxiliary pathways, and these are the description. See, we have 27 peptide count. Okay, so wow, it's a good. We have to globally related markers, and they have very good p-value score with a 27 peptide count. Again, the complementary for but they have a lower p-value and good change. And a false positive rate for the same. So also in the box plot, we can clearly see that the it has clearly identified the idiopathic pulmonary fibrosis very well. So these novel ones are very good identifying it with a very good confidence interval. We can see the 95% interval and a cutoff of this. So yeah, this is very good. This is a bar chart, and this is another one that I think they used here IPA. This is a box one. This is for activation, showing the what is activating. Yeah, this is very good. So we clearly see that they did volcano plot hierarchy analysis, and they got the p-value of around 0.05 over the 0.5, and complement those protein also were mentioned. I will see that, but the haptoglobin markers are very good folding as well as protein isolations. So I will say brilliant. But you can say identify potassium biomarker. We can clearly see in the what. Let me show. Uh, Here the scope plot. This is a partially square discriminant analysis in which the control group is here, and the what do we say for that? What's its name? Yeah, the haptoglobins. These are marked, and the thing which comes in the between in them, we will say that yeah, this control group and the idiopathic are combinedly same. The control group be anything which is inducing the idiopathic, which is inducing an idiopathic. So yeah, it's brilliant. With from that, again the p values are taken out. You can see haptoglobin is coming exactly 0.05, and with a peptide count of 27. So that's a brilliant word. 0.05 coming up within that is very good. And unique peptide also. So those peptides which are not even seen. So the part one is also liver significant dissolution. So we have these unique peptides, unique haptoglobins, which say that yeah, idiopathic pulmonary fibrosis happening. So very good paper. Mike's did very good. And in conclusion, I have to say that haptoglobin protein related is a promising biomarker. We need further research in it, further validations, whether these all things are happening or not. and literature review also, that what more research is coming out in this idiopathic, this haptoglobin related, uh, working as a biomarker for idiopathic pulmonary fibrosis. And complementary oxidative pathways are crucial for idiopathic pathogenesis and further and explore. So the key findings are the haptoglobin related biomarker, and its dissolution, it causes dissolution in the complement pathway and oxidative stress pathway in IPF patients. So what I learned from the manuscript is that importance of comprehensive proteomics analysis in identifying disease biomarkers, and significance of pathway analysis in understanding disease mechanisms. So whenever we are doing analysis, so protein analysis, it's a very difficult part. It does not go like what do we say. You take the serum, you bring it out in the lab, and you have to identify. You can just do volcanic plots. You have to identify particular analytical tests and to identify a particular protein. For example, in the paper, these guys, they did volcanic plot also. They did partial discipline, as well as they did a clustering heat analysis. Clustering is just to identify all the markers, but the correct thing was this core plot, and this is one of the advanced statistical analysis, which when we have a huge set, then this can be very well used to get that narrow it down to the particular what we say markers to the control group, and they easily show it in the log value. Now this is work on a plot. This is work. This is a work on a plot XY log values, and they got it. And this one is actually, I think, this figure 4 is for actually, I think. Yeah, this is ROC curve, area under curve. This is the volcano plot. Volcano plot shows us the what type of here haptoglobin here is. Let me zoom in. We can get the haptoglobin related markers coming out of this. These guys are these markers are not changing, but this haptoglobin related markers also changing. So haptoglobin related markers is getting better, and these guys can also be taking CPX3 and CBB240. So these are coming as a minus negative. These are getting more mark. So yeah, even the volcano plot also confirms it. But whenever you conduct the statistical analysis, see partial least discriminant analysis is way better. It clearly narrows it down, and volcano plot will validate your findings. So yeah, whenever choosing protein analysis, the test should be very carefully chosen. And significant of part medicine resistance. Yeah, the same. See, whenever we go with complement pathway system and oxidative pathway system, this complement pathway system, these systems are also working different disease mechanisms. Also, for example, they told you about idiopathic pulmonary fibrosis. Now let us go with something else. Let us say idiopathic arrhythmia, or let us say any infectious disease due to any virus. Then also the same pathway, same oxidative pathway is also there. So the pathogenesis also has to be understood very well. Currently, the it is idiopathic pulmonary fibrosis pathogenesis is not well understood. They say that due to some idiopathic nature, the fibrosis occurs. There's necrosis. No, not necrosis. For the first, the tissue gets damaged and then it occurs. So this is pathogenesis also needs to be understood very well, along with the proteome. So for the future research findings, research plans, if possible, let us go with large independent cohorts like control case study between they use around 37 individuals. Here they said talked about here they took 36 individuals. So we'll need a larger group of around, let's say, 100 to get to know that longitudinal study to understand biomarkers. So let us longitudinal study says that go with go with 5 10 cohorts, then to 20 cohorts into a longitudinal studies. and many therapeutic applications of identified pathways. So currently, this we took the pathway which was using the haptoglobin markers. So we should also see for any drug discovery also which can happen, identifying the markers, any blockers which use this as. Currently, I think haptoglobin markers literature uses it diagnostic purposes. But in future, we can say that they can become therapeutic purposes also if a blocker is found out. So I think I've summarized. Let me summarize in the end. First, some of the points which I would suggest. First is that uh, my paper is on liver-free plasma protein identify, and we want to identify haptoglobin proteins, which are a candidate marker for idiopathic. The paper, which is the original paper, let me show you the original paper. The original paper is this, and the co-author for this is Mayank Saraswat, and he has extensive research in the respiratory disease, especially in the proteomic analysis as well as biomarker discovery. And what is idiopathic pulmonary fibrosis? It's a chronic progressive disease, and it's very essential and very necessary to diagnose early so as to get effective treatment. And one of the novel technologies to use for diagnosis is proteomic studies. Upon the proteomic, normal proteomics such as our own haptoglobin markers shown promising results. So its main objective was to potential of new identifying haptoglobin markers as for biomarker IPF, as well as understanding the pathogenesis. So these proteins take new pathogenesis pathway. So even we are going with pathogenesis also. And also again 36 individuals. We use advanced techniques as well as identifying significant proteins which are different from case control groups. Um, Label-free proteomics means those proteins we have not been labeled previously in any studies, as well as advanced analysis. Like here, this advanced is the partially discriminant methods. Mm, oh God, let me change it here. Yeah, the results are that the haptoglobin identifies the potential marker. Again, you can go with the paper, and you can clearly see the what is the how the cluster map is formed, or partially how the volcano load describes that. Along with haptoglobin marker, the p-value is coming 0.0. It's a very good marker. Haptoglobin markers is very good, and it also shows a new oxidative pathways as well as complement activation system pathway for IPF pathogenesis. And haptoglobin marker is a very good potential marker. So it comprehends proteomic analysis into the and pathways is very necessary. More research should be done for understanding this mechanism. Future plan conduct longitudinal studies as well as to do the activity in larger independent groups and explore therapeutic approaches. So with this, I'd like to end my presentation. I hope something could be understood. And with this, I end. Thank you very much.